very atypical, and they're kind of diagnostic of cells having suffered very severe DNA damage. And a dicentric is when they've got these two primary constrictions, and this again is an abnormal structure that forms in chromosomes after particular mutagenic events. So what happens is that after a cell has detected a genotoxic insult, like it's been exposed to radiation, it'll initiate repair processes. So this is complicated biochemical event, but it's initiated by proteins detecting the DNA damage and then assembling big protein complexes around the sites of the damage to repair them. This is what our lab is particularly interested in. So here, I don't know if you can actually, can you make out any nuclei here? Can you see? No, okay. There are some, okay? So about the same, you can make them out here. Okay, you can see the same density of cells displayed in both micrographs. But here you can see the irradiated cells have begun to form these, we call them nuclear foci, because they're assembling proteins together into the molecular complexes that are going to repair the DNA. And then, as you can see, over time, this damage signal diminishes because the cells are carrying out the DNA repair. So we've subjected all the cells to a strong amount of radiation. After about 5 minutes, even after radiation, they begin to say, oh, we've got to repair our DNA. And then over time, as the DNA is repaired, these complexes disperse because the DNA damage has been resolved. So there's active and cellularly controlled processes that cause the repair of the DNA damage. And this is effectively what keeps your cells and your genomes intact in the 30,000 genes that you have in the 3,000 million bases of each cell of your genome. So this is a very important cellular process. Now, it usually works, but sometimes if cells are subjected to a certain amount of DNA damage, abnormal things can happen. The repair process mightn't always work properly. Okay, so one of the things we discovered around about 2002 was that after certain levels of DNA damage, the normal nice bipolar mitotic cells, this is where they're going to divide in the two directions that I showed you, the Lily trying to do earlier in the talk, can form crazy looking mitotic structures like this. You can see here, this is a very, very abnormal structure. You've gone from a nice bipolar, you're going to try and separate the chromosomes in two directions. Now, how many poles are there here? 1, 2, 3, 4, 5, 6, 7. Okay, but this cell has only got 2 copies of every chromosome, so when it tries to divide into 5 or 7 or did anyone say 9? I estimate about 7, but we might have seen all the poles here. Obviously, all these daughter cells are going to get an insufficient amount of the chromosomal material, so this is going to end fairly badly for the cell whenever it tries to undergo mitosis.